So today I will present a work I've done during my PhD, during a stay abroad at Columbia University at Professor Molly Chervosky's lab, which has the title "The Evolution and Coevolution of PRDM9 Across Vertebrates." So during this talk, I'm going to give you a brief background of what PRDM9 is and its importance in recombination among vertebrates. I'm going to introduce you the data, the methods, and results we have used in this work. And then at the end of this talk, I'm going to draw some conclusions. So as evolutionary biologists, we can think that there are two sources of genetic variation that can introduce genetic variation into the population. So the genetic variation that is present here today between us humans comes from first from mutation. So deletions, inversions, translocations, or point mutations that are caused by environmental or non-environmental factors can affect and change the DNA sequence and therefore introduce genetic variation. And then another source of genetic variation is also homologous recombination. So in sexually reproducing species, there is a moment in meiosis, specifically in meiosis 1, in which the DNA homologous chromosomes coming from the different parental lineage, they cross and they exchange DNA. During this process of crossover, there is the possibility of creating new combinations of alleles, and those alleles are then possibly introduced in the population. These new combinations of alleles are then shaped by selection and genetic drift, and that's how species evolve. Homologous recombination is also important for fertility because it allows the proper alignment and the proper segregation of chromosomes during meiosis. This proper alignment and segregation makes us avoid the possibilities of aneuploidies and then therefore ensure the success of gametogenesis, which therefore is going to affect, of course, the fertility of species. So the places where recombination occurs defines the recombination landscape of a given species. And the pace by which recombination occurs defines the recombination rate of that given species. In 2010, 3 different independent groups have realized or they have found that the recombination landscape and the recombination rate in humans is dependent on one peculiar gene, and this gene is called PRDM9. This gene encodes a protein that is characterized by 4 different domains. So it has the CRAB domain, the SSXRD domain, a SET domain, and a zinc finger array. In 2010, they realized that there were motifs along the human genome that were associated with the zinc finger array of this protein, and those motifs were also associated with increased rate of recombination, which then later were called hotspots. So hotspots are increased rate of recombination. So the domain architecture of this gene is very important, and it defines 2 things in a given species. It first defines the recombination landscape. So for a species in which PRDM9 is not present or truncated like birds or dogs, there is a fold increase in recombination rate in genomic-like features such as transcription site sites and CPG islands. As you can see here in the graph, there is a fold increase of recombination rate. But for a species in which PRDM9 is complete, it has all the domains, then this fold increase is not observed. Instead, the increase is observed in regions in which the zinc finger binds in the genome. So another important another thing that it defines the domain architecture of the gene defines is the evolutionary pace of the zinc finger array, of its zinc finger array. So in species for which we have a complete PRDM9 with all the domains, then the zinc finger is rapidly evolving. So there is like a high turnover of its zinc finger along evolution. However, for species in which there is a truncated PRDM9 with one or two missing domains, then the zinc finger is not rapidly evolving, suggesting that PRDM9 is not actually targeting double-strand breaks along the genomes of those species. So how does PRDM9 work? So it first binds to a motif through its zinc finger array, and then it uses the set domain to methylate the surrounding nucleosomes of that binding motif. And then it uses the CRAB and the SXRD domain to interact with other proteins. And then after the region is marked by PRDM9, then the gene or the protein SPO11 is then recruited and then to initiate the double-strand break. It's not well known how SPO11 is recruited, but once recruited, then many other proteins are then recruited for the process of crossover, which in the end is gonna turn on non-crossover or crossover resolutions. So our main goal with this project was to characterize PRDM9 classification across vertebrates, and then also to identify genes that were co-evolving with PRDM9. And I would like to thank all these people, especially Molly, for the work that I'm presenting today. So we first decided to look at RNA-seq data from 4 different species for which we had no other source of data to characterize PRDM9. So we took tissue samples from muscles, testes, liver, from 2 reptiles, so anole and fence lizard, and 2 fish, Mexican cave and Atlantic herring. And then we basically did the de novo assembly of those transcripts. And then we tried to search, doing a reciprocal blast search of PRDM9 within those transcriptomes. The pipeline is as follows: library preparation, sequence, de novo assembly using Trinity, PRDM9 identification using the blast search, and then transcript quantification. So the results are shown here. We didn't find PRDM9 expression in the reptile species, and therefore we couldn't characterize the PRDM9 architecture. We found PRDM9 expressed in the 2 other fish, but only the set domain was found in the Mexican cave, while in the Atlantic herring, we found both crab and set domain. So what is the expected effects of those results in the recombination landscape of the species? So for those species in which we were unable to find PRDM9, recombination rates are expected to be elevated near promoter-like features such as CPG islands and transcription start sites, just because those are regions with more chromatin accessibility. So SPO11 breaks whatever is accessible. And then we also expect to find a stable evolution of recombination hotspots. And for this second species in which we found an incomplete PRDM9, a similar pattern is observed similar to that in reptiles in which PRDM9 is absent. And then for this species, we believe that actually this fish has a complete PRDM9. So the hotspots are, the events of recombination are likely observed in hotspots, similar to those species in which a PRDM9 is complete, like most mammals. So we also wanted to date these specific losses of reptiles. So what we did was to plot those losses along the vertebrate phylogeny, specifically along the reptile phylogeny. And then we tried to find what was the closest species for which a PRDM9 was complete. So this species here, which is the birded dragon, is a species that contain a complete PRDM9, and the loss must have occurred between in the common ancestors of anoles and fence lizard, and it should have occurred between 99 and 157 million years ago. So we wanted to be able to identify genes co-evolving with PRDM9. And we were able to at least classify 4 species based on RNA-seq data. We combined that RNA-seq classification with whole genome sequence search and with protein sequence database search. So we looked at RefSeq and the whole genomes in order to characterize the rest of the vertebrates that we could. So we characterized the PRDM9 domain architecture across 379 vertebrate species. And this amount here is plotted here. As you may know, birds have lost PRDM9. So of course we didn't find PRDM9 in those species. We also don't find in wolves, or we find only a truncated version of PRDM9 in wolves, and we do not find them in dogs. And then in the fish clade, there is like, it's very interesting, PRDM9 has been lost multiple times in the fish clade. So we wanted to understand if we were able to detect genes co-evolving with PRDM9. So our strategy here was to try to transform PRDM9 status in a binary trait. So for all species in which we had an incomplete or a partial or a loss of PRDM9, we treated those species as zeros or black balls here in this slide. And then for species in which a complete PRDM9 was present, we treated them as ones or red circles here. And then we tried to correlate, to search for genes that were coincidentally lost with the loss of function of PRDM9 across vertebrates. So for that, we restricted our search to a data set in which we only had meiosis-related genes. So we restricted our search of PRDM9 against a given gene to a set combined of 3 sets. The first was genes involved in meiotic recombination in mammals, and then also included in this search the top 1% of genes co-expressed with PRDM9 in mice spermatogenesis based on single cell analysis. And we also include the genes associated with recombination rate phenotypes in humans. And then for this entire data set, we did a gene orthology search and then computed the presence and absence matrix of those candidates across 339 species, and then compared that presence and absence matrix against our PRDM9 absence and presence matrix. So the results are shown here, in which we found very few genes. So this is the QQ plot of the phylogenetic independent contrast of PRDM9 and the many different genes. And we can see here the both red and gray dots are significant results using 5% significance threshold. And the red dots ones are genes that are actually co-expressed with PRDM9 in spermatogenesis. You can see here the expression of PRDM9 and some of the other genes. And what I want to highlight here today is the coincident loss of PRDM9 with 2 other genes, ZCWPW1 and ZCWPW2. We are using many more species than this specific plot that I'm showing here, but just to give you an idea of the coincident losses that occur, that we have seen to occur between the loss of function of PRDM9 and the loss of the other gene. It's very interesting that we found the ZCPW1 because 3 different groups in 2019 have found that the ZCPW1 is able to read both epigenetic marks laid by PRDM9. And they also found out, based on the mouse knockout of this gene, that PRDM9 recruits this gene to its binding site across the genome. But this gene does not help to position the breaks, the double-strand breaks themselves. So it has nothing to do with positioning double-strand breaks by SPO11. But it is essential for the double-strand break repair and the chromosome pairing, and therefore essential for fertility in mice and possibly in mammals. So could the other gene, ZCWPW2, be interacting with PRDM9? So the ZCWPW2 is a paralog of ZCWPW1. It also has both domains that can read both epigenetic marks, H3K4 methylation and H3K36 methylations of PRDM9. It has both structures that are found in the PW1, and it also, based on residual conservation analysis, I also found that those domains are the most conserved regions of the protein structure across vertebrates. So ZCPW2 could possibly be the missing link between PRDM9 and SPO11. One could think that PRDM9 first methylates the nucleosome surrounding its binding site, and then ZCWPW2 read those methylations, use both domains. And then once it reads, then it recruits SPO11, and then SPO11 comes and do the double-strand break initiation. So to conclude, we found, based on RNA-seq data, a complete loss of PRDM9 in 2 reptile species that must have occurred between 157 and 99 million years ago. Very few genes, including ZCWPW1 and ZCWPW2, showed non-independent losses with PRDM9. Both W1 and W2 have domain structures that can read the epigenetic marks laid by PRDM9. ZCPW1 has been recently found to be essential for double-strand break repair and chromosome pairing, but not for the initiation of double-strand break. And functional analyses are necessary to understand the role of ZCPW2 in meiosis, but it could be the possible missing link between SPO11 and PRDM9. With that, I would like to say thank you. Yeah, and I'm excited to hear questions. Thank you.